AS Level Biology 9700, Paper 2, Variant 1, October November 2025. And in this video, we will discuss question number 3. And question number 3 is the lock and key hypothesis and the induced fit hypotheses are used to describe the interaction of enzymes and their substrate. So describe one similarity and one difference between the lock and key hypothesis and the induced fit hypothesis. So first of all, we have to write down the similarity and the difference, then difference between these two, having 2 marks, one for each. That is one for similarities and one for difference. So first of all, lock and key, according to the lock and key hypothesis, that active site is rigid, fixed shape that is perfectly complementary to the substrate before they even meet. So first of all, we will write its similarity. In both hypotheses, the substrate binds to the enzyme's active site to form an enzyme substrate complex. In both hypotheses, the substrate binds to active site to form enzyme substrate complex. So in both cases it happens. Both models rely on the principles of complementary shapes. So meaning the substrate must fit the active site for the reaction to occur. The substrate must fit to the active site. Now we will write the difference. So the difference is, in lock and key hypothesis, that active site is a rigid fixed shape that is perfectly complemented to the substrate before they even meet. Whereas in induced fit, the substrate enters the active site, changes shape slightly to mold more tightly around the substrate, imposing the fit and putting strain on the substrate. So in lock and key, they just work like lock and key, but in induced fit, shape is little bit altered to fit the substrate onto it. So we will write all this on the given space as I'm writing on the screen. So it will take more space than writing on a proper paper. So first of all, according to the lock and key, active site is rigid, fixed shape that is perfectly complementary. Complementary to the substrate. To the substrate before they even meet. But in induce fit model as the substrate enters the active site, the active site changes shape. Changes shape slightly to mold around the substrate. So these are one similarity and one difference between them. Now moving towards the next question, part B. And in this, we can see an equation, figure 3.1. So, which has been written over there. Phosphorylase enzymes can catalyze the synthesis of starch and the breakdown of starch in some plant tissues. A reaction catalyzed by starch phosphorylase is shown in figure 3.1. So you can see the part of the starch molecule, glucose 1-phosphate and starch phosphorylase will act as an enzyme. So at the end, there will be the formation of phosphate ions, and then this is the starch molecule. You can say it will join this carbon will join with it, and then there will be the release of phosphate ions with it. So a student carried out an experiment to study the synthesis of starch by phosphorylase found in potato tissues. The student was provided with the solution E extracted from potato tissue. The extract was filtered to remove all the starch grains. Extract E contained biological molecules from the potato tissue, including phosphorylase, but no starch. Iodine solution was used to confirm that starch was not present in extract E. State the color, state the color observed when iodine solution was added to the sample of extract E. According to the iodine solution, students, you know that iodine solution is normally a yellow-brown color. So if starch is present, it would react with the iodine to turn a distinct blue-black color. In positive, it will change. Yellow-brown color will be changed into blue-black. So in this case, when there is no starch, then what would happen? There will be a negative result. It means it will not change. So it will remain the same color, which is the color of the yellow brown. This no change if no starch is there. So the original color will be of the iodine will remain there. Next question. The student added a small drop of a dilute starch solution and a solution of glucose 1-phosphate to a test tube containing extract E. Samples of the reactions mixture in the test tube were removed every minute and a drop of iodine solution was added to each. The student used a calorimeter to measure the absorbance of the solution in each sample. The results are shown in figure 3.2. So you can see the results over here in figure 3.2. Here you can see that on x-axis they have taken the time, on y-axis they have taken the absorbance. So here in this case, you can see that a linear curve has been obtained. That is, as time is increasing, the absorbance is also increasing. So it means in this graph shows a steady increase in the absorptions over a 12-minute period. You can say it was for about 12 minutes. So the absorbance is increasing. That is in this way. You can see a little bit linear curve has been obtained in this case. So we have to explain these results in figure 3.2. So let me write it down over the given space. That first of all, I would like to explain, then I will write it over there. As as the progress, the enzyme in the extract T converts the substrate into starch molecule. The small drops of dilute starch added the starch act as a primer for this reaction. So iodine solution is used as an indicator. In this case, when iodine reacts with starch, it forms a blue-black complex. Already told you in previous question. So as more starch is produced over time, the blue-black color of the samples becomes more intense in a colorimeter because if there is more starch, so more color will be obtained. So the darkened solution absorb more light, resulting in the inner increase in absorption shows in the graph. So the constant positive gradient indicates that the enzyme is working at a steady rate, that at neither the substrate nor the enzyme has become a limiting factor. So we have to write all this for about 3 marks. So I'm going to write over here the enzyme in extract E converts the substrate into starch molecules. So this is the way as you will write in your exams. The small drop dilute starch added at the start acted as primer. Iodine will be used as an indicator. As more starch is produced over time, the blue-black color of the sample becomes more intense. Darker solution absorbs more light. More light resulting in linear increase in absorbance. It shows it shows that the enzyme is working at a steady rate. Well, it was part 2. Now moving towards the next part of this question. After 12 minutes, the student added a solution containing phosphate ions to the reaction mixture. The student continued taking samples every minute, adding iodine solution to each sample. So after 12 minutes, so it will take place after 12 minutes. So the absorbance, after that, the absorbance of the solution in each of these samples was measured. The results showed that the absorbance decreases over time. Suggest why the absorbance decreases as the experiment was continued. So we can better understand that as the time passes, the enzyme in the mixture is breaking down the starch into smaller sugar molecules, maybe the glucose or maltose. Uh, so in many biological practicals, we can say that the enzyme phosphorylase phosphate ions act as a substrate or activator. They trigger the breakdown of starch. So as starch is used up, there is less starch available to react with the iodine. So therefore, the blue-black color becomes lighter and the measured absorbance decreases. Already we have discussed that absorbance is increased when the color of that solution is dark. Darker color will absorb more, uh, absorbance will be increased in darker colors. But here you can see the color is decreasing. So it means it becoming lighter. So it means that the starch is broken down by the enzyme. So there is less starch to react with the iodine, leading to a lighter color, lower absorbance. So I'm going to write this, which I have explained to you. The starch is being broken down by the enzyme. So there is less starch to react with the iodine, leading to a lighter color. In other words, we can say that the enzyme, there is no enzyme left behind to work with. So it was all about this question. It was of only 1 mark. Now moving towards the next part of this question. So muscle cells contain glycogen phosphorylase. So figure 3.3 shows the effect of caffeine on the activity of glycogen phosphorylase at different concentrations of substrate. So our student concluded, you can see the two graphs. On x-axis, the concentration of substrate has been taken. On y-axis, the glucose phosphorylase activity has been taken. So, and the key we should observe, it is a circle inside a dot when there is no caffeine. When there is caffeine, they have put a cross mark. So we will go with these two curves. You can see when there is without caffeine, you can see the maximum V maximum. But with caffeine, you can see the V maximum has been decreased for this. So a student that caffeine act as a non-competitive inhibitor of glycogen phosphorylase. Explain how the result in figure 3.3 support this conclusion. Now move to this. You can see that without caffeine, V maximum is high. So with caffeine, the V maximum is lowered. So it means the caffeine will act as a non-competitive inhibitor. And you know, what does it mean? That is, what is non-competitive inhibitor? So non-competitive inhibition. So a student has concluded this. Explain. So non-competitive inhibitor is that inhibitor which binds to an allosteric site. So this changes the shape of the enzyme, rendering it less effective or inactive regardless of how much substrate is present. So I'm going to write this explanation. Without caffeine, the enzyme actively reaches a maximum. Actively reaches a maximum velocity, V maximum, of approximately 195 attributary units. So with caffeine, with caffeine, the maximum, the V maximum is drastically reduced to approximately 95 arbitrary units. So because the V maximum is significantly lowered and cannot be restored by adding more substrate. So the inhibition must be non. So it was all about this question. We have completed this question. So please like, share, and subscribe my channel. Thanks for watching.